Dear students, this is our 12th lecture on bioorganic chemistry and continuing with mechanism of enzyme action. In this lecture we will see the action of lysozyme enzyme on different types of substrates. Lysozyme is an enzyme which hydrolyzes polysaccharide chains, and it breaks certain bacterial cells by cleaving the polysaccharide chains which build up their cell walls. The most common example in case of lysozyme is studied in the form of hen egg whites, and this bacteriolytic properties of hen egg white lysozyme they were described in 1909 by Russian scientist Lysenko, whereas in 1922 Alexander Fleming gave the name to this enzyme as lysozyme because he found that this is an agent in the mucus and tears that destroys certain bacteria, because it was an enzyme which caused bacterial lysis, and because of this bacterial lysis this was given the enzyme lysozyme. This was given the name lysozyme. Now what happens is, in case of bacteria cells, they are surrounded by rigid strong walls of peptidoglycan. Peptidoglycan is a copolymer of two sugar units. One is called as N-acetyl muramic acid. This is N-acetyl muramic acid, which is called as NAM, and other is N-acetyl glucosamine. This is N-acetyl glucosamine. So this is NAG, this is NAM. This is NAG, N-acetyl glucosamine. This is N-acetyl muramic acid. This is NAM. So all these repetition of this NAG and NAM will form the bacterial cell wall in the form of polysaccharide, and these NAM and NAG, they are joined by beta 1,4 glycosidic linkage. This is beta 1,4 glycosidic linkage which is present, and this lysozyme hydrolyzes the glycosidic bond between C1 of NAM. NAM means N-acetyl muramic acid. This is N-acetyl muramic acid, and C4 of NAG. This is C4 of NAG. This is C4. This is C1 of NAM. This is C4 is of NAG. So this will hydrolyze this beta 1,4 linkage, not this beta 1,4 linkage. So between NAG, C1 of NAM and C4 of NAG. NAG means N-acetyl glucosamine. NAM means N-acetyl muramic acid. So hydrolysis of these ether linkage, that is beta 1,4 linkages, will take place, not these beta linkages. So this will break from here and we will get the dimers like this, NAG and NAM, NAG and NAM. Two molecules will be formed by hydrolytic cleavage of this. So mechanism can be a carbocation mechanism, like here there are two active sites. One is glutamic acid 35 and aspartic acid 52. So this aspartic acid 52, glutamic acid 35. This is present in the form of acid. This is present in the form of carboxylate ion. So in the first step, the glutamic 35 act as a general acid. This will act as a general acid. General acid means this proton can be abstracted by the substrate. This oxygen as a nucleophilic center will abstract this proton this will become O positive and H will get attached to this. We will get this thing, and this glutamic acid will acquire the negative charge like this. Then movement of this lone pair to this place and breaking of this carbon oxygen bond between NAM and NAG will take place. N-acetyl glucosamine, N-acetyl murmuric acid and acetyl glucosamine. So this carbon oxygen bond will break at this place in the second. So the protonation makes the leaving group a weaker base, hence this will become a better leaving group in the second step. Then in the third step, this part is removed. This is the first product. This is a positively charged entity, and now this aspartic acid 52 will attack as a nucleophile on this carbon in the presence of a nucleophile like water. So nucleophilicity of this water will be increased by this glutamic acid 35, and this will attack as hydroxide ion at this carbon, and we will get the second product like this. So this is how in the third step this product has been removed. In the fifth, this product will be removed. So this is how the hydrolysis will complete with the hydrolysis of NAG and NAM. Then another type of mechanism which was given in 2001 is SN2 mechanism. Here the two consecutive direct bimolecular nucleophilic displacement reactions take place, and we know that in case of SN2 there is an inversion of configuration, and when there are two SN2 reactions are taking place, that means there will be two inversions of configuration, and net result will be that the product will be formed with retention of configuration. So here in the first SN2 reaction, this aspartic acid active site will attack this carbon to break this carbon oxygen bond, and this will acquire negative charge, and at the same time this will obstruct this proton, and we will get the first product eliminated like this. And then when first product is eliminated, this has formed a covalent bond with the substrate. This enzyme active site has formed a covalent bond with the substrate. In the next step, this will interact with the nucleophile hydrolytic water, and nucleophilicity of this nucleophile will be increased by glutamic acid active site. This will obstruct this proton, and this will attack as a hydroxide ion, and this covalent bond between substrate and the enzyme active site will break, and the second part of the product will be removed. And you can see there are two SN2 reaction. Here inversion of configuration take place 1, here inversion of configuration will take place 2, and overall there will be retention of configuration in the both the products which are formed, and this type of mechanism is called as SN2 mechanism. Thank you very much.